I think if everyone sits down, we can get started. Because if we get late, then you'll have less time for lunch and posters, and that's an important component. So please, everyone, have a seat. You know, every year in our meeting, we have two marquee lectures. They are our main lectures, and that's to honor our co-founders. As we know, 52 years back, in 1969, APA was founded by three great scientists, great pancreatologists. One of them, we are fortunate, Dr. Bill Go is here with us. The other two were Paul Webster, who could not be here today, and the third one is late Dr. Frank Brooks. Dr. Brooks was a very eminent gastroenterologist, pancreatologist from University of Pennsylvania. He was the chair of gastroenterology there, been past president of AGA, but more important, I think, is that he was a co-founder of APA and president of American Pancreatic Association. So in his honor, we have a Frank Brooks lecture every year. This year, it's my privilege to introduce our speaker, Dr. Miklos Sahin. Not that he needs much introduction. Miklos is really a world-renowned expert in area of pancreatic disorders, pancreatitis, both acute and chronic. And his main focus for last about 20 years or so has been on genetic risk factors for pancreatitis. I met Miklos, well, he finished his MD and PhD from Budapest, and then came as a postdoc at Scripps Institute in La Jolla. And from there, I think his first faculty job was in Boston University, and that's where I had a privilege of meeting Miklos. So that's about almost 20 years back now. And since then, we've become a good friend, great friends. But more importantly, I learned a lot from Miklos in his research. Miklos really was and is still a hardcore biochemist who really we used to call that he has a factory of making mutant digestive enzymes. He has every mutant of almost every digestive enzyme in the pancreas in the lab, and then he will study their biology. Eventually, he moved and he realized that he needs to do that you cannot just study enzymes and pancreatitis in test tube. So the models which we use are the animal models. So he created several different new strains, new models of mouse pancreatitis, which has really changed the landscape of what we know about pathobiology of pancreatitis. So from Boston University, he was recruited by our current president, Dr. Hines, to UCLA about 2 years back now, or something like that, and where he has really established his lab and really gets started. Needless to say that Miklos has many publications, I think about 150 and counting, and most of them are really in very good journals. Most of them are in Nature journals, Nature Genetics, Nature this, Nature that. And I think he has published few in gastroenterology and once in a while in gut. So we are very proud of his publications. And to publish all these things and do all this, you need a lot of money. So Miklos has many, many grants from NIH and other places. But more importantly, Miklos has been a great president for our organizations about 5 years back. And I am so proud of your accomplishment. Welcome, Miklos. Look forward to your presentation. Except it was a requirement that for this lecture one has to wear a tie, and Miklos refused that. So I don't know, maybe we have to do something about that.
Thank you so much. That was just way too kind. And it was, as Ashok said, we met when I was at Boston University for 17 years, and that's when we met. And Ashok was at Worcester, he used to go out every Tuesday. I learned a lot from him. And I always tell the story that he asked me, can you come? So when our routine was, I would go out on Tuesdays and he would buy me, breakfast at the cafeteria and we would talk. And then one day he asked me, can you come on Wednesdays? Because they have a discount on breakfast on Wednesdays. And then I recently moved to UCLA, and I'm really grateful for the welcome and the support of Joe Hines and then Agi Hirschberg, which helped us with the transition. I wasn't sure if Ashok will introduce Frank Brooks, so I copied it from the APA website, who Frank Brooks was. I came up with this really great title, which more and more I like. What I like to just discuss briefly a little bit is how hereditary pancreatitis changed our field, the discovery of the genetic basis, how it stimulated new research and led to some very interesting insight, how pancreatitis in general develops. And the reason we do this actually is to understand mechanism, because once you understand mechanism, you can design preclinical models, you can test therapeutics, and eventually deliver new treatments for patients. And if we speak of mechanisms, we actually had a mechanism once presented in a Frank Brooks lecture, and this is we all stand on the shoulder of giants, and this is one of, the late Mike Steer is one of the giants in pancreatology, who gave the very same talk I'm giving right now. And 98, maybe he gave it 97, published in 98. And the content of his talk I distilled to this one slide, where he describes how pancreatitis begins. And there's some injury, there's a secretion block in the S inner cell, and two enzymes, trypsinogen and catepsin B, get co-localized. One is normally in zymogen granules, other is in the lysosomes. They should not really meet that much. And then catepsin B activates trypsinogen and you get pancreatitis. And if I take it down further to the molecular level, this is what this model looked like. Again, catepsin B activates trypsinogen to trypsin, and trypsin would activate other proteolytic zymogens. You get a protease soup, and that initiates inflammation. Now, these different proteases come in different flavors. These are called isoforms, and I'm indicating the human isoforms on this particular slide. For trypsinogens, and I will focus on trypsinogens in this talk, the gene symbols are PRSS1, 2, and 3 in the humans, which are not really intuitive, but that's how they name them. You notice a little green arrow going from trypsin to trypsinogen. So trypsin can also activate trypsinogen, and we call this process autoactivation. In the original model, it's actually this process hasn't been indicated because catepsin B was the activator. At least that's what we were thinking at the time. And taking it one level down and showing the activation mechanism, the way trypsin or catepsin B activates trypsinogen is cleaves the activation peptide, which is this unique N-terminal sequence, which has this characteristic polyaspartic motif, and the cleavage is after a lysine residue, and which is shown in red. And the mechanism of activation is always the same. Now I'm showing the activation, which is the pathological activation inside S inner cells. Normally trypsinogens are activated by an enzyme called enterokinase in the gut, but that's not expressed in the pancreas, so we're not going to be discussing that. So this seemed like a very easy to understand model. It had a lot of evidence, coming from not only from Mike Steer's group, but many other groups over the years. But things started to change actually in the late 90s. And this was the first set of papers which came out in 96, when 3 independent groups identified the chromosomal location of the gene responsible for hereditary pancreatitis. Now, hereditary pancreatitis has been known since the 50s as an autosomal dominant disorder, which is very rare. In certain families, it pops up. And at this time, they identified the chromosomal location. Now, at 1996, if you knew the chromosomal location, that was fairly useless information, because we did not have a human genome sequence. Except the same year, somebody sequenced the T-cell receptor beta locus, and they happened to find trypsinogens in this particular locus scattered among the immune genes. So this really gave these groups, and particularly one group, Dave Whitcomb's group, a candidate gene to look at. And in 96, Dave published this seminal paper which identified R122H as the causative genetic change in hereditary pancreatitis. And I cannot really, I'm biased of course, but I cannot really think of any other discovery that had such an impact on our understanding of how pancreatitis begins mechanistically and also on the clinical side. So I'm showing 2 more papers on this slide, because these 2 more papers already foreshadowed what was about to come, an explosion of the field. There was a whole bunch of new mutations discovered in trypsinogen, and this N29I is another clinically more frequent trypsinogen mutation. But there was like 50 others found eventually, and a whole other set of new genes were identified, and many mutations in those genes. So it was really remarkable how this whole field of genetic pancreatitis evolved. Again, today we will focus on trypsinogens. Another aspect of this slide is that it brought in new people into the field. So if you look at the paper on the bottom, which is a paper from Leipzig by Niels Teich, and Niels Teich would never study pancreatitis normally, but with this new field, new technology, a lot of people moved into pancreatitis research, looking at genetics and association with pancreatitis. So one of the new people who moved in was a young enzymologist, yours truly, who got interested reading these papers. I said, this could be testable. This is an enzymatic problem. You take the enzyme, put in the mutation, let's see what happens. It wasn't that simple, obviously. And this is one of the first studies we did. We had a model, cathepsin B activates trypsinogen. You have trypsinogen mutations. It stands to reason mutations will change how cathepsin B activates trypsinogen. And this was in collaboration with Markus Lerck's group at the time. And we were a little surprised and disappointed. If you look on the left side, that the clinically common mutations do not change cathepsin B-mediated activation in the test tube. Not only that, but if you look at some of the other mutations shown on the right side, some are actually block cathepsin B activation. So you have a mutation like this D22G, which is not activated by cathepsin B, but causes hereditary pancreatitis. Now, Markus Lerck said, trypsin is protective. But it wasn't the case eventually. But that was a very provocative hypothesis. But when you look at the same subset of mutations, these actually stimulated autoactivation tremendously. And this is just spontaneous conversion of trypsinogen to trypsin by autoactivation, in which trypsin activates trypsinogen. This was a very attractive hypothesis. The problem with this was that many of the clinically frequent mutations did not do this. So it took us another 5 years to identify that human trypsinogens are actually under the control of this enzyme called Chymotrypsin C, which proteolytically regulates the activation process. And Chymotrypsin C does 2 different things. The main impact, it degrades trypsinogen. If you degrade trypsinogen, it cannot activate, so that's a protective function. Paradoxically, there's an activation stimulation function of Chymotrypsin C also. And the way to picture this is shown on the next slide. If you look at the yellow, that's an autoactivation curve. And if you run this in the presence of chymotrypsin C, you see that the initial rate of activation is stimulated. And this is by trimming the activation peptide, actually. But the major effect of chymotrypsin C is the protective, the degradation effect. And there's a reason for this stimulation, because the degradation actually requires a little trypsin. So chymotrypsin C helps generate a little trypsin, which eventually helps it to degrade trypsinogen. So once we had this recognition that activation of trypsinogen is regulated by chymotrypsin C, and we retested the mutations in this context, now we see huge differences. The clinically significant mutations actually stimulate autoactivation dramatically when tested in the context of chymotrypsin C. And to put it back into the previous slide, this context, this shows a number of mutations, which looks like the phone book, I understand, so don't look at the particular mutations, but show the mechanistic insight we gained. Many of the clinically more frequent mutations, like R122H, they block degradation by chymotrypsin C, and that thereby stimulate activations. There's a subset of mutations which actually directly stimulate activation. Those are shown on the left side, and these are like in black. And there's a very interesting subset, and the most interesting is the A16V mutation, which is fairly common, which hijacks the stimulatory activity of chymotrypsin C and stimulates the stimulation of activation. So this, what you don't see in this whole picture is cathepsin B. Because having tested a very large number of trypsinogen mutants, we found only one where cathepsin B showed some stimulatory effects. The genetic data did not support any participation of cathepsin B. In fact, if the father of cathepsin B, Marcus Lerch himself, ruled out cathepsin B as a genetic risk factor in a study. So at this stage, we felt that, as Ashok already pointed out, that we need to move to some mouse models to validate these findings, if this is really the case. And I'd like to thank Bo and G from yesterday's talk, where he actually highlighted many of our models and his models, which are very similar in phenotype. So in the first model, which was published in 2018, after almost 5 or 6 years of work, we did a very simple thing, just model autoactivation of trypsinogen to trypsin in mice. And we call this model T7D23A, and I explain this in the next slide. So T7 is the mouse cationic trypsinogen. Mouse has 20 trypsinogen genes numbered 1 through 20. 7 happens to be cationic trypsinogen. And the mutation is changing the aspartic acid before the activation site lysine into an alanine. And this mutation is analogous to the D22G mutation found in hereditary pancreatitis. Note that the numbering is shifted by 1 in the mouse gene with respect to the human gene. This mutation dramatically stimulates autoactivation. In fact, this kind of stimulation we do not see with the human mutations, but we were interested in conceptually model, the effect of autoactivation. And indeed, when you look at the phenotype of the mouse, you see a dramatic phenotype. About age 3 weeks, the mice develop acute pancreatitis, and acute pancreatitis rapidly progresses to early chronic pancreatitis and end-stage disease with adipose replacement in this particular model. So it not only conceptually confirmed that autoactivation can drive disease onset and progression, it also showed that acute and chronic pancreatitis can be a sequence within the same disease process, driven by a single genetic change. We were curious what happens if you do a more moderate model, where still driven by trypsinogen autoactivation, but not as drastic as a 50-fold increase. And so it's a very similar model. It's K24R. And the mutation is now the activation site lysine is mutated. And there's an exact same mutation in hereditary pancreatitis called K23R. So these particular mice do not get any spontaneous phenotype. And if you look at in the test tube on the left side, so this mutation stimulates autoactivation about 5-fold, not 50-fold as the other. And if you challenge the mice with one injection of cerulein hyperstimulation, you see there's more intrapancreatic trypsin activation compared to the control Black 6 strain. And consistent with this increased trypsin response, you see a more severe pancreatitis. This is showing just edema. You can actually see the pancreatic lobules as they separate on the lower right side. You look at histology, you see, again, much more edema, more inflammatory cells, and overall a more severe pancreatitis. Not only that, but in this model, pancreatitis actually spontaneously progresses after one injury of cerulein. So after a single episode, if you look at the control strain, it quickly recovers within a few days. But the trypsin mutant mice, develop, chronic changes, and I'm showing you the first 3 days. But even if you go out to 2 weeks, 1 month, 3 months, the pancreas is still not normal, and it takes almost, and even at 3 months, you see adipose replacement where cells died. So there's a progressive chronic-like phenotype driven by trypsin autoactivation. So taken together, these two models not only validate all the genetic and biochemical data we had seen before and demonstrate that trypsinogen autoactivation caused by mutations is responsible for disease onset and progression, but they also are great preclinical models where you can test interventions. Each model has its advantages. Obviously, a spontaneous model is more like a human situation. But this model, and I'm going to show you on the next slide, you can actually experimentally handle this model much better. If you look at this is pancreas weight as a function of time. Starting with an acute episode, you see at the black dots, you see an increase in pancreas weight due to edema, and then you see rapid atrophy, and the pancreas recovers very slowly. So you can do very nicely synchronized experiments with this model, either looking at the acute phase or the chronic phase, and you can intervene at various time points. So this particular model has that advantage. And by the way, all the models are freely available if anybody is interested in experimentation. So before I show you my last slide on the future perspective, how we should really move into drug testing, I have to come back to Cationic B. Because this model was such a solid model in the field, and we failed to generate any evidence which would actually validate this in the human setting. So we decided to approach this from the aspect of trypsinogens. And this is the work of originally Alexander Demchak and then Andrea Geis, both of whom are attending this meeting. We decided to change trypsinogen in a manner which it cannot auto-activate anymore, but it's activated by Cationic B. And then in the paper which is quoted there is a biochemical paper just to identify what the best mutation would be. And the two examples shown, the mutations are actual mouse lines we generated using these mutations. So in one of the mice, trypsinogen is activated normally by Cationic B. In the other mice, there's increased activation. So the expectation was that maybe in that mouse we will see spontaneous pancreatitis. We did not see any spontaneous phenotype in these. When Andrea looked at trypsin activation as induced by cerulein, in the normally activating mutant, you see normal activation. In the increased activation mutant, you see increased activation. So the biochemical phenotypes were actually replicated inside the mouse. The big surprise came in the next experiment when you look at pancreatitis severity, and you don't see any difference. So regardless of the level of intrapancreatic trypsin activation, you get the same control phenotype back. And on the left side, you see some histology examples, and the right side shows pancreas mass as a readout for edema, but you get the same thing for amylase or MPO. So this was very surprising, and the way we interpreted this is, indeed, there is catepsin B-mediated trypsin activation, but it's completely irrelevant to the disease pathogenesis, and it doesn't determine disease severity. And when we look at the literature, we actually find that we're not the first to say this. And here's a long list of papers. These are all mouse models, many of them published by eminent authors from the audience, where there's a discrepancy between trypsin activation and disease severity. In some of the papers, like the catepsin B knockout or the trypsinogen knockout, published by Markus Lerck and Ashok Saluja, there's no trypsin activation. You still get good pancreatitis, maybe a little improved. In the other two models, the catepsin R knockout or that strange colocalization model, there is increased trypsin activation, and you get no change or you get decreased severity. So based on this data, we put it to you that I think it's time to forget catepsin B as a good disease target, and we can argue about it after the talk. And then focus back on trypsin. And with the final slide, I'd like to highlight that we're in a very good position now to really go back to drug testing. There's been a lot of effort initially, the past 20 years, to look at trypsin inhibitors in pancreatitis. We did not have the right models. So much of the data published is not relevant, I think. And we should revisit this with having now very good, preclinical models. We should also, think about gene therapy. And these discussions with Sohail, I realized that adeno-associated virus expresses things for a long time in the pancreas. And chymotrypsins may be exploited as a protective mechanism, although we think of those as enzymes, which may be, but it looks like in terms of pancreatitis, it protects. So these are just two possibilities, but we definitely have to continue with this. So instead of showing you a skyline of Los Angeles, I'd like to leave this slide on, and then thank you for the attention, and we're happy to discuss during lunch or afterwards.
Thank you, Miklos, for a very, very nice, clear history of trypsinogen and catepsins in pancreatitis. We have 2 minutes for questions. So short questions, just a couple of them, and then we'll move on.
Thank you very much for this wonderful presentation, Miklos. We've been arguing about this back and forth, and collaborating on this back and forth for roughly 20 years now. And it took a while till we understood that chymotrypsin is the key regulator, and because we couldn't explain so many mutations that were outside the box of activity. Let me argue one more point. I think it's very, very well established that cathepsin play a role in trypsin activation and trypsin degradation, the L with the C, B, and also the D. I would interpret your data differently. Maybe trypsin activity up to a certain point is immaterial for the development of pancreatitis. And that's what Ashok showed by knocking it out and still getting some way of pancreatitis. But what is it? What would you speculate if trypsin is clearly the factor for inducing genetically determined pancreatitis, but trypsin activity may not be the key factor? What other property of trypsin is it that may be the driving force of pancreatitis, at least in humans? Or is it the space where trypsin activity is? Outside the cell, inside the cytoplasm, inside vesicles. We were always focused on vesicles, but maybe that's the wrong location.
I'm thinking outside right now, so we have no evidence, but based on many of our models, I'm thinking interstitial, presence is more important than intravesicular presence. And for many of the studies come from the Cerulein model, and it looks like Cerulein just through CCK-mediated pathways. Maybe that's what Ashok says, that you get NF-kappa B activation regardless of trypsin, if I interpret correctly.
Julia, please have a short questions. We're going to have less time.
I leave out the cadapsin B and trypsin discussion. So the question I'm asking, or I'm interested in, is you showed us a model of chronic pancreatitis, but this model resembles atrophy. There was some inflammation, but no stroma whatsoever. And the question is, what is the clinical endpoint we are studying if you use this mouse as for drug development? So is that a clinically relevant model? Is there pain? Is there malnutrition? So because if we want to take it to a clinical, use it as a clinical model, then there might be other aspects to be...
Yeah, those are excellent points. Just we don't fully understand that fibrosis is why it's not prominent in these models. But as Boan G showed actually in some of his recent work, that you can use these models because atrophy is a great readout. It's a very convenient laboratory readout. And if he treats mice with dabigatran with similar models, you get very robust healing, and you can follow it by histology and atrophy. Obviously, it's the whole fibrosis issue that might require different models.
Dr. Roach?
So, Miklos, excellent talk. Congratulations. So my question is, do the models develop exocrine pancreatic insufficiency and diabetes? If not, why not?
No diabetes, that I don't fully understand. They do develop actually insufficiency, both models, different degrees. But weight gain stops at one point, and in the more acute, where there's a cerulein induction, there's a more acute loss in weight, and you can see that actually.
Last question, Dr. Bonji.
Okay, two quick questions. First one is in human PRSS1 mutation causes pancreatitis. By why so far at least the transgenic mice of human PRSS1 mutation doesn't cause spontaneous pancreatitis? Number two, I would like to repeat Ashok's question. We also trypsin activation can cause acinar cell death and inflammation. How?
So the reason you don't see pancreatitis in your model, it may be the level of expression, because you add on another trypsin, PRSS2, and now you see spontaneous pancreatitis. So we believe that the total level of trypsinogen expression and the propensity to autoactivate, it has to reach a certain level to see spontaneous disease. Otherwise, but they're still very sensitive to cerulein-induced disease, right?
Yeah.
And what trypsin does and how it leads to inflammation, I think that's the next important question, as was pointed out yesterday too, and we can speculate. I'm back to PAR receptors actually in my mind, but because there's nothing new under the sun, right?
Okay, thank you.
Thank you very much. So now in order to, Dr. Hines, can you please come up? We're going to take a picture. So in order to celebrate your very distinguished lecture, Frank Book lecture, American Pancreatic Association is going to present you this plaque, which hopefully will adorn your office.
Well, thank you. Thank you.
Okay, I think we need to go down for it.
Okay.